Okay, let's start. Number 1. The Starchild skull. You are holding a skull in a storage room in Chihuahua, Mexico. The bone is wrong before you've had time to think about it. Too light. Almost half the weight it should be. You turn it in your hands and the eye sockets are too shallow, not slightly recessed, but dramatically flat, barely indented at all. The brow ridge is absent. The frontal sinuses are gone. Every anatomically modern human who has ever lived has frontal sinuses, the air pockets behind the forehead that develop during childhood without exception. This skull doesn't. Run your finger along the interior surface where you would normally feel smooth bone. It's layered with fibers woven into the matrix like the inside of a carbon composite shell. No known primate has bone built this way. The cranial volume, measured by seed-fill displacement, is 1,600 cubic centimeters. The human average is 1,200. More brain space than a standard human, half the bone weight, a structure that doesn't appear in any known species. This is what the Starchild skull is. And that's just what you can see with your hands. The skull was found in the 1930s by an American girl hiking in Mexico's Copper Canyon region. She discovered 2 skeletons buried together in a tunnel in the hills near Chihuahua, one fully human, one radically different. The skulls passed through private hands for decades before reaching Lloyd Pye, an author and researcher, in 1999. Pye commissioned formal scientific testing and spent the remaining 14 years of his life pursuing answers. In that time, the skull accumulated more documented physical and genetic analysis than almost any anomalous artifact in the historical record. The physical findings were consistent and disturbing at every stage. X-ray diffraction and electron microscopy confirmed the presence of fibers distributed throughout the bone matrix, not found in any known human skull, not found in any known animal skull on Earth. Eye socket depth measured 0.6 inches compared to the standard 1.4 inches in Homo sapiens. No frontal sinuses. The inion, the bony prominence at the rear of the skull that anchors neck muscles, is absent. The mastoid processes behind the ears are flat, where they should protrude significantly. These are standard anatomical features present in every human, including those with known cranial deformities. Everyone is missing from the Starchild skull. That is not pathology. That is a different construction. DNA testing at Bold Labs in Vancouver in 1999 extracted mitochondrial DNA inherited through the mother and identified it as haplogroup C, consistent with a Native American female. A human mother. But the paternal nuclear DNA presented a different result. Sequences recovered from the skull's nuclear genome did not match any entry in the human genome database. Not Homo sapiens. Not Neanderthal. Not any other known hominid. A 2010 follow-up using next-generation sequencing attempted more complete recovery. The results confirmed the earlier finding segments of the recovered nuclear genome had no registered match in any species. The analysis was never submitted for peer review. Critics cite that constantly. What they don't discuss is that no independent laboratory has been able to explain the anomalous sequences either. They matched nothing then. They still match nothing now. The mainstream explanation is hydrocephalus, a condition causing abnormal fluid accumulation and cranial expansion. Hydrocephalus explains the enlarged size. It does not produce bone that weighs half of what bone should weigh. It does not create woven fiber structures in the matrix. It does not erase frontal sinuses that develop in every human hydrocephalic or otherwise without exception. And it does not produce paternal DNA, with no match anywhere in the genome of our species. Lloyd Pye died in 2013 with the analysis unfinished. The skull remains privately held. No major institution has formally accepted it for independent testing. The sequences that matched nothing have never been explained. No one has tried very hard to explain them. Who was the father? The DNA doesn't say. Neither does history. Number 2. The Paracas elongated skulls. You're in a museum in Ica, Peru. The shelf in front of you holds 40 skulls. Every single one is wrong. Each cranium is dramatically elongated, not slightly misshapen, not compressed by burial, but extended several inches at the rear and tapering into a narrow peak. The bone is dense, intact, structural. The cranial volume in the largest specimens, measured using seed-fill displacement, reaches 2,800 cubic centimeters. An average adult human skull holds 1,200. You are looking at skulls with more than twice the cranial capacity of a normal human head. You cannot create that with cloth. You cannot wrap an infant's soft skull tightly enough, for long enough, to manufacture 2.5 times the brain volume of the person next to you. You cannot add bone that was never going to grow. And there is something else, something that doesn't appear in any textbook on cranial deformation, because it cannot be explained by it at all. In 1928, Peruvian archaeologist Julio Tello began excavating the Paracas Peninsula on Peru's southern coast. He uncovered a network of underground burial chambers holding more than 300 mummified individuals, many with dramatically elongated skulls. The burials dated to between 3,000 and 2,000 years before the Common Era. Preservation was extraordinary. The desert climate maintained skin, hair, and tissue on many specimens. The hair on numerous individuals was red or auburn, not dark, not black, not the hair color associated with any documented pre-Columbian Andean population. The color was confirmed as natural, not post-mortem oxidation of dark hair, not dye. Genetic red hair pigmentation present in people who should not, by any conventional understanding of that region's population history, have had it. The mainstream explanation for elongated skulls in ancient cultures is cranial binding documented across South America, Africa, and Mesoamerica, in which an infant's soft skull is bound during the first years of life to reshape it. Binding works by redistributing bone mass as the skull grows. This is real, documented, and the accepted explanation for most elongated skulls in the Americas. But binding has a hard biological ceiling. It moves mass. It cannot create mass. Every bound skull from other Andean cultures measures within the normal human cranial volume range of 1,000 to 1,350 cubic centimeters. The largest Paracas specimens exceed 2,800. That is not redistribution. That is additional biology. There is also the matter of the bone plates. Every Homo sapiens has 3 cranial plates, the frontal plate and 2 parietal plates, fused by sutures that close progressively as the brain finishes developing. Multiple Paracas specimens have 2 parietal plates, not 3. This is not a deformity. A deformity is an abnormal version of a normal structure. These skulls have a different plate architecture. You cannot produce a different plate count by wrapping cloth around an infant's skull. The sutures run in different positions. The foramen parietal, a small opening at the top of the skull, is enlarged consistently across the specimens. These are not variations. These are structural differences in fundamental bone organization. In 2014, researcher Brian Forster coordinated DNA testing on hair, skin, and bone samples from 5 Paracas mummies. The samples went to a geneticist who agreed to analyze them on condition of anonymity, citing professional risk. The preliminary results showed that the mitochondrial DNA haplogroup recovered from the samples matched no known Native American lineage, no known Asian lineage, and no known European or African haplogroup. An unknown haplogroup, a genetic line that either doesn't exist in any modern population or has never been catalogued. When the preliminary findings circulated, the researcher received threats. Full publication has not occurred. Peruvian government restrictions on access to the museum specimens have tightened since 2014. The official position remains cranial deformation. The plate count anomaly and the cranial volume measurements do not appear in that official position. Cranial binding moves bone. It cannot create bone. Whose genome was that, in the mitochondrial sample that matched no known lineage? And why does the full publication keep failing to appear? Number 3. The Ubaid lizardmen. You're an archaeologist in 1919 at Tel al-Ubaid in southern Iraq. You are excavating a layer of soil that predates the Sumerian civilization itself, older than writing, older than the wheel, older than any of the structures we call the cradle of civilization. You pull a clay figurine from the dirt. It is female. It is nursing an infant. The skull is elongated and flattened. The eyes are large, almond-shaped, and slit like a reptile's. The facial structure is angular and non-human. The sculptor rendered the skin with texture, not human skin texture. Scales. And the figure holds the infant the way a mother holds a child. Tenderly, securely. Whoever made this was not depicting a monster. They were depicting someone they knew. H.R. Hall excavated Tel al-Ubaid near the ancient city of Ur in 1919 on behalf of the British Museum. He recovered structures, pottery, and figurines belonging to what archaeologists would later identify as the Ubaid culture, a civilization that flourished from roughly 6,500 to 3,800 years before the Common Era, well before the Sumerians emerged. Further excavations across southern Mesopotamia and into modern Kuwait, Bahrain, and eastern Saudi Arabia recovered the same type of figurines from sites separated by hundreds of miles. The total count across the Ubaid cultural layer now exceeds 5,000 individual specimens. Two distinct types appear in the record. Some figurines are clearly human normal facial proportions, round eyes, standard skull shapes. The other type has elongated skulls, almond or slit eyes, angular facial structures, and in many cases a textured skin surface that reads as scaled. Both male and female versions exist. Female figures nurse infants with the same reptilian anatomy. Male figures hold scepters or objects associated with authority. They are not depicted as servants or as dangers. They are depicted as rulers and as mothers. They hold social position. They are in charge. This is what separates the Ubaid figurines from ordinary religious iconography. Ancient cultures depict monsters as threatening, distorted, clearly supernatural. The Ubaid, non-human figures, appear in domestic and authority contexts. The artistic tradition is not one of fear, it is one of representation. The Sumerians, who inherited much of Ubaid culture, later produced the Sumerian king list, a document that opens with the line, "When kingship descended from heaven." The first kings named on that list have reign lengths of tens of thousands of years and are identified as semi-divine or non-human in origin. The Ubaid figurines predate those texts by 3,000 years. They show non-human beings in positions of rulership, represented with the same artistic consistency the Sumerians used for their historical kings. The mainstream interpretation is that the figurines are ritual or shamanic objects, animal totems, fertility symbols, water spirits. The problem with that interpretation is specificity. Every other Ubaid figurine depicting a human looks distinctly human. The reptilian figurines are not stylized deviations. They are consistent across sites separated by 500 miles, across a cultural period spanning nearly 3,000 years. The eye shape is the same. The skull elongation is the same. The scale texture is the same. A 3,000-year artistic tradition maintaining one consistent non-human anatomy across multiple disconnected sites is not an abstract spiritual concept. It is documentation of a consistent subject. After the 2003 invasion of Iraq, the Iraq Museum in Baghdad was looted. An estimated 15,000 artifacts disappeared. The Ubaid figurine collection, including many specimens that had never been formally catalogued, was among the losses. The British Museum holds a portion. The remainder is scattered through private collections whose contents have never been made public. A full comparative analysis of the complete Ubaid corpus has never been published. What were the Sumerians remembering when they wrote that kingship came from heaven? And why do the oldest representations of those kings look like that? Number 4. The Lovelock Cave Giants. The Paiute Indians have told this story for generations. In the ancient past, a race of red-haired cannibals occupied their land. The Paiute called them the Sitaka. After years of war, the Paiute and their allied tribes drove the Sitaka into a cave, piled brush across the entrance, and set it alight. The red-haired ones burned. The story was told to every generation that followed. It was told to the early American settlers. It was written down. Anthropologists called it legend. Then, in 1912, miners digging commercial bat guano from a cave 22 miles south of Lovelock, Nevada, broke through a sealed layer of earth and found what was inside. The red-haired mummies were exactly where the story said they would be. Lovelock Cave was excavated professionally between 1912 and 1924, first by Llewellyn Loud from the University of California, then by Mark Harrington of the Museum of the American Indian. The teams recovered remains from at least 60 individuals alongside thousands of artifacts, duck decoys made of tool reed, woven baskets, weapons, and organic material that carbon dated to between 2,000 and 3,000 years before the Common Era. The hair on multiple mummified remains was red or reddish-brown. Laboratory analysis confirmed the color was natural, not post-mortem darkening, not dye, not oxidation of black hair over time. Genetic red hair pigmentation. In a pre-Columbian Nevada cave, among a population whose surrounding regional neighbors produced no such coloring. Loud and Harrington recorded their findings in field notes taken during excavation. Those notes described several individuals whose skeletal measurements indicated heights of 6.5 to 7 feet. In a region where the average Great Basin Native American stood approximately 5 feet 3 inches, individuals of that stature were exceptional. When Loud published the formal academic monograph Lovelock Cave, issued by the University of California Press in 1929, the size descriptions were revised. The largest individuals were characterized as large, but within the normal range. The original field notes say something different. The published paper is what archaeologists cite. The field notes sit in archives. In June of 1931, the Nevada Review Minor published 2 separate reports of additional skeletal finds near Lovelock. One skeleton measured 8 feet 2 inches. Another was described as close to 10 feet. These specimens were never formally accessioned into any academic collection. No peer-reviewed analysis exists for them. They appeared in the historical record for a few column inches and then effectively ceased to exist. The Smithsonian Institution acquired a significant portion of the formerly excavated Lovelock remains. Researchers and Paiute tribal members who have subsequently attempted to access the largest specimens for direct measurement or genetic testing have encountered access restrictions whose rationale has never been publicly explained. The skull morphology of several Lovelock individuals shows prognathism, a forward projection of the jaw structure that is inconsistent with the facial architecture of surrounding Native American populations. This is not a feature of cranial binding or nutritional stress. It is structural. Repatriation requests by Paiute tribes under the 1990 Native American Graves Protection and Repatriation Act have complicated modern genetic testing. The tribal position is that the remains belong to their ancestral territory and should be returned to the earth. That position is understandable. It also means the specimens that would definitively answer the size and genetics questions are no longer available for testing. The conversation closed before it could open. The Paiute story described the Siteka. The cave confirmed the red hair. The field notes confirmed the height. The published papers said within normal range. What would we find if someone actually measured the ones in Smithsonian storage? Number 5. The Atacama skeleton. In 2003, a treasure hunter excavating the ghost town of La Noria in Chile's Atacama desert found a tiny humanoid figure wrapped in white cloth behind an abandoned church. It was 6 inches long. Its skull was cone-shaped and dramatically elongated. Its eye sockets were large relative to its face. It had 10 ribs, not 12. Its bones showed the cortical density and growth plate development of a child between 6 and 8 years old. It was not a doll. It was not a figurine. It was biological flesh, cartilage, and bone, preserved almost perfectly by the driest air on Earth. And it had been alive. Not briefly, not for hours. Alive long enough for its bones to develop density that only comes with years of living. The skeleton, nicknamed Ata, passed through the antiquities market and was eventually acquired by a Spanish businessman named Ramon Navia Osorio. In 2012, it reached Dr. Garry Nolan at Stanford Medical School's Department of Microbiology and Immunology. Nolan was one of the world's leading experts in flow cytometry and human genomic analysis. He subjected the specimen to full genomic sequencing, the most rigorous scientific examination it had ever received. The results were published in 2013 in Genome Research. The mainstream scientific community has been trying to close the case ever since, and it won't stay closed. Ada was human. The mitochondrial DNA placed her within haplogroup B2, a lineage associated with Native American populations of South America. That was not the problem. The problem was what the nuclear DNA produced. Full genomic sequencing identified 64 novel mutations, genetic variants never before observed together in any individual in the entire human genome database. Many of these mutations affect bone development and skull morphology. The specific genes involved include COL1, A1, COL2, A1, and FLNB, all associated with skeletal dysplasias. But no single known dysplasia and no combination of known dysplasias produces all features observed in Ada simultaneously. The 10 rib count is a precise example. 11 rib pairs appear in certain rare conditions. 10 rib pairs are not a documented outcome of any known skeletal dysplasia in any patient, anywhere in the medical literature. In 2018, Stephen Binns published a paper in Genome Research arguing that Ata was a stillborn fetus or premature infant with multiple coexisting bone disorders. The scientific mainstream accepted this. Case closed, but the bone density analysis was never adequately addressed. The growth plates, the regions of developing cartilage at the ends of long bones that calcify progressively during childhood, show a developmental pattern associated with a child who had been alive for years, not hours. A stillborn fetus does not develop cortical bone architecture that a radiologist reads as age 6 to 8. A stillborn fetus is not 6 inches tall with a fully formed skeleton, 10 functional ribs, and a completely developed, elongated cranium. These are not subjective readings, they are measurements. Dr. Nolan, after the 2018 paper, did not fully concede. He has stated publicly that the simultaneous presence of 64 novel mutations warrants continued investigation and that Ata's genomic profile remains one of the most unusual datasets his laboratory has produced. The remains were returned to Chile amid legal disputes about whether they had been legally exported in the first place. Their current location under Chilean antiquities law is contested. The physical specimen that could be re-examined is no longer available for re-examination. 64 mutations, never observed together in any recorded human. In a body whose bones say it lived for years, in a desert that preserved it for 1,000. The case was closed before the questions were answered. What exactly is a human when its genome matches nothing ever recorded? Number 6. The Denisovan bones. In 2008, Russian archaeologists excavating Denisova cave in the Altai Mountains of Siberia found a piece of bone. It was part of a finger. The fragment was 1.2 inches long, smaller than your pinky fingertip. They sent it to Svante Pääbo's team at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany. The analysis took 2 years. When the results came back in 2010, they ran them again. The DNA didn't match Homo sapiens. It didn't match Neanderthals. It didn't match any species in the entire known fossil record of the human family. From a fragment smaller than a child's fingertip, they had found an entirely unknown species of human, one that no one had suspected existed, and the genome it left behind was more complex, in some respects, than ours. Pääbo's team published the discovery in Nature in 2010, naming the new species Denisovans after the cave. Further specimens emerged over the following decade. A molar tooth so large that the Russian archaeologist who initially found it assumed it was from a cave bear. A toe bone. A partial jaw, recovered from a monastery supply cave on the Tibetan Plateau in 2019 by Fahu Chen's team, more than 1,600 miles from Denisova Cave, confirming the species had ranged across Asia far more broadly than anyone imagined. From all sites combined, the physical remains of fewer than 8 individuals are known. That is everything. That is the entire physical record of a species that lived across Asia for hundreds of thousands of years. The genome they left behind told a richer story than the bones. Denisovan DNA showed greater genetic diversity than Neanderthal populations, suggesting a deeper and more geographically spread lineage. Their genome also contains sequences with no close match in any known primate, living or extinct, indicating that the Denisovans themselves had interbred with something else, something older, something that has not yet been found in the fossil record. The Denisovans are not only a mystery. They are a clue, pointing toward a deeper mystery that the bones beneath our feet have not yet produced. The most concrete trace the Denisovans left is in the bodies of people alive today. Populations in Melanesia and Papua New Guinea carry up to 6% Denisovan DNA. Mainland Asians and Native Americans carry roughly 0.2%. But the most precise Denisovan contribution exists in Tibetans. A gene variant called EPAS, one which regulates how red blood cells process oxygen and allows the body to function normally at extreme altitude, is derived entirely from Denisovan interbreeding with an ancestral human. Population. Tibetans have it. Han Chinese at lower elevations don't. Without that Denisovan gene, sustained life above 13,000 feet produces chronic altitude sickness, reduced fertility, and shorter lifespans. The Denisovans gave us the ability to live on the roof of the world. Their genome was different enough from ours to provide something ours couldn't build on its own. The Denisovan molar is enormous, larger than any Neanderthal or Homo sapiens molar in the fossil record. Researchers estimate that an individual with a tooth of that size had a body substantially larger than modern humans. That is inference, not measurement. There are no long bones, no skull. No reconstruction is possible. We can sequence their genome with precision. We cannot picture their face. After more than a decade of the most advanced paleogenomics ever conducted, we still don't know what they looked like. That gap in the physical record is not explained by preservation conditions alone. Fossils of contemporaneous species survive. The Denisovan physical record, for a species that ranged across an entire continent, is almost nothing. What else was here alongside us? And what does it mean that they helped build us? Number 7. The Anunnaki tablets. In the British Museum's collection, among roughly 130,000 clay tablets recovered from ancient Mesopotamia, most excavated by Austin Henry Layard from the Library of Ashurbanipal at Nineveh between 1849 and 1851, are texts that describe the origin of the human species, not in metaphor, not in cosmic poetry, in operational language. They describe beings called the Anunnaki, a Sumerian term meaning those who from heaven to earth came, who modified the genetic material of an existing primate to create a being capable of sustained labor. These tablets are between 4,000 and 6,000 years old. They predate the Book of Genesis by at least 1,500 years. They are physically real. They are catalogued. The question is not whether they exist. The question is what they are documenting. The key Sumerian creation texts include the Enuma Elish, a Babylonian epic on 7 tablets, and the older Atrahasis, which draws on traditions several thousand years older still. The Atrahasis is explicit in a way that makes scholars uncomfortable. It describes the Anunnaki as a class of beings who arrived and established civilization. After a period of time, the text records, the lesser Anunnaki rebelled against the labor of digging irrigation canals. The solution stated directly, not implied, was to create a lesser being to perform the work. The creation procedure involved mixing the blood of a slain divine being with clay. The Sumerian word translated as blood is team, a term that also carries the meaning of intelligence or essence. The word translated as clay also means earthly biological substrate. Zecharia Sitchin, a linguist whose translations began appearing in 1976, read these passages as literal descriptions of genetic engineering and was dismissed by the academic mainstream as sensationalist. But the question his reading raises doesn't go away. The Atrahasis text describes a procedure. Materials are mixed. Failures occur. Success is achieved. A specific outcome is produced. That is the language of process, not poetry. What mainstream Assyriologists agree on, the Anunnaki texts describe non-human beings who predated humanity, who came from somewhere external, and who were causally responsible for the creation or modification of human beings. The dispute is interpretive myth versus literal history. Samuel Noah Kramer, whose foundational work at the University of Chicago remains the standard scholarly reference on Sumerian civilization, treated the texts as sophisticated mythological narrative. He also acknowledged that the Sumerians themselves treated the Anunnaki as historical beings, not symbolic ones. The Sumerian king list, which transitions seamlessly from pre-flood rulers with reign lengths of tens of thousands of years into historically verifiable Sumerian kings, makes no distinction between myth and history. There is one piece of physical evidence that cannot be dismissed as translation dispute. Human beings have 46 chromosomes. Every other great ape chimpanzee, gorilla, orangutan, bonobo has 48. At some point in human prehistory, 2 ancestral primate chromosomes were fused into a single chromosome, producing what we now call chromosome 2. The fusion point is identifiable. In 1991, J.W. Ido and colleagues confirmed the fusion in a paper published in the Proceedings of the National Academy of Sciences. The mainstream explanation is a spontaneous chromosomal rearrangement in a single individual whose descendants eventually carried the new configuration to fixation throughout the ancestral human population. That sequence of events is not impossible. The probability calculations for a single random mutation becoming fixed in an entire population are extremely low and require a specific combination of small population size and strong selective advantage. Neither has been demonstrated for chromosome 2. The fusion exists. The explanation for it does not satisfy the mathematics. The Sumerian texts describe a directed modification of primate genetic material. The human genome shows a modification of primate genetic material that appears directed. The tablets are real. The chromosome is real. The gap between them requires an explanation. If the Anunnaki are myth, why does our chromosome count match the outcome they described? Number 8. The Tocharian mummies. In 1979, Chinese archaeologists excavating the Taklamakan Desert in the Xinjiang region of western China found mummies they were not prepared for. The bodies were 3,000 to 4,000 years old, preserved almost perfectly by one of the driest climates on Earth. The faces were angular, long noses, prominent brow ridges, deep-set eye sockets. The hair was blonde, auburn, and reddish-brown. The frames were tall. The most famous specimen recovered from the site of Lulan, and dated to approximately 1,800 years before the Common Era, was a woman researchers named the Beauty of Lulan. A male from the site of Churchin stood 6 feet 6 inches tall with a red beard and wore woven wool trousers in a tartan pattern. He looked European. He was 2,000 miles from Europe. And he and his people had been in that desert for thousands of years, before any documented contact between East Asia and the Western world existed. The Chinese government restricted access to the Tarim Basin mummies for decades. Foreign researchers were denied genetic samples through the 1980s and most of the 1990s. Victor Mayer at the University of Pennsylvania became the first Western scientist to conduct systematic study, publishing his findings in 1998. The genetic analysis confirmed what the physical features suggested. These people were not ancestral to the Han Chinese. Their mitochondrial DNA fell into haplogroups H, V, and U lineages, strongly associated with West Eurasian and European populations. Y chromosome analysis showed a mix of Siberian and Iranian ancestry. No genetic marker matched the populations that geographically surrounded them. The textile evidence added another layer. The Churchin man's clothing was woven wool, dyed and patterned using a twill weave identical to Bronze Age European textile techniques. The tartan pattern on his trousers matches weaving traditions found in Bronze Age, Central Europe, and Scotland. No Bronze Age East Asian textile tradition used this technique. Their material culture was as foreign to the region as their genetics. In 2021, Vagesh Narasimhan and colleagues at Harvard and the University of Texas at Austin published full genomic analysis of 13 Tarim Basin mummies in Nature, the most comprehensive genetic study of the specimens ever conducted. The findings settled the question of whether these were migrants from the West and opened a far deeper one. The mummies were not migrants, not from Europe, not from the Eurasian steppe. They descended from a lineage geneticists call the ancient North Eurasians, a ghost population known only through the partial genetic traces it left in later populations, with no identified homeland, no associated archaeological culture, and no living descendants anywhere on Earth. The Tarim mummies were the last isolated survivors of this lineage, preserved by desert conditions while the rest of their kind vanished without physical trace from the historical record. The ancient North Eurasians fed partial genetic contributions into early Native Americans, certain Northern European populations, and some South Asian groups, always as a minor secondary component, mixed into other lineages. Only in the Tarim Basin did they survive alone and unmixed for thousands of years. They practiced burial rites with parallels in Vedic Indian tradition, early Iranian culture, and Bronze Age European burial customs, 3 separate cultural traditions with no known vector of transmission between them. They were in a desert that should have been impossible to inhabit, practicing customs from civilizations they had no documented contact with, preserving a genetic line that no longer exists anywhere else in the world. A ghost population with no living descendants, in a desert that kills most things, practicing rituals from 3 separate civilizations simultaneously. Where did the ancient North Eurasians come from? They left traces in our genome and then ceased to exist. Number 9. Homo naledi. In 2013, experienced cavers exploring South Africa's Rising Star cave system were directed toward a passage the survey maps hadn't fully charted. To reach what lay beyond it, they descended a vertical shaft and then squeezed through a crack in the rock. The crack was 7 inches wide at its narrowest point. They had to exhale completely before pulling themselves through sideways. On the other side of that crack was not one skeleton. It was 15. The chamber floor was covered with hominin remains, multiple individuals, different ages, a population. The researchers selected to retrieve the bones were partly chosen by body size because no one larger than a specific frame could physically fit through the slot. When paleoanthropologist Lee Berger at the University of the Witwatersrand saw the first photographs, he knew immediately the anatomy was unlike anything in the fossil record. Not deformed, not unusual. Structurally, unlike any known species in the human family tree. Berger and his colleagues published the discovery in the journal eLife in 2015, naming the new species Homo naledi, meaning star in the Sesotho language. The anatomy produced a mosaic that had no equivalent among known hominins. The brain measured approximately 500 cubic centimeters, comparable to the earliest members of the Homo genus from almost 2 million years ago, about a third the size of a modern human brain. The shoulders were rotated forward in a configuration suited to tree climbing. The fingers were long and curved, showing wear patterns indicating sustained arboreal activity. And yet the feet were modern. The arch, toe alignment, and proportions were nearly indistinguishable from those of Homo sapiens. The wrists showed a precision grip configuration associated with tool use. The teeth were small and refined, more advanced in some respects than species with far larger brains. No known evolutionary lineage produces that combination. An ape's shoulder architecture on a body with modern human feet, primitive brain volume in a creature capable of a precision grip. The anatomical mosaic implies either a radically unusual case of convergent evolution or a history of deep interbreeding between distantly related lineages, neither of which has a clear precedent anywhere else in the hominin. Record. The 2015 announcement assumed, based on the primitive anatomy, that Homo naledi was ancient, perhaps 2 to 3 million years old. In 2017, John Hawkes and colleagues published dating results in eLife. The Dinaledi chamber remains were between 236,000 and 335,000 years old. Homo naledi was not ancient. It was alive at the same time as early Homo sapiens in Africa. Two profoundly different human species, sharing a continent, with no clear documented evidence of contact, one with a modern brain-building symbolic culture, one with a brain one-third that size, doing something in a cave that took a great deal of deliberate effort. That effort is the part researchers argue about most fiercely. The Dinaledi chamber has no surface access. Nothing falls or washes into it. The route requires the 7-inch squeeze and a long vertical descent. There is no evidence of predator activity that would accumulate bones. The assemblage includes individuals of multiple ages, which argues against an isolated accident. The only physical explanation consistent with the evidence is that Homo naledi deliberately carried its dead into that chamber. If true, this is the oldest documented deliberate mortuary behavior by any non-modern human species performed by a creature with a brain a third the size of ours, indicating that the concept of death as something requiring a response predates the brain capacity we assumed was necessary to have it. It was alive when we were alive. It was burying its dead when we were. It looked nothing like us. How many other species were sharing this planet with us that we haven't found yet? Number 10. The Guanche mummies of the Canary Islands. The Canary Islands sit 62 miles off the northwestern coast of Africa. When Genoese explorers made contact in 1312 and Spanish conquistadors arrived in force in the late 14th century, they found an advanced people already established on the island's farming, building in stone, maintaining a complex social hierarchy. The Spanish called them the Guanche. The Guanche had no boats. This is not a simplification. Contemporary Spanish colonial records, corroborated by the complete absence of any watercraft or maritime technology in the Canary Islands archaeological record, confirm that the Guanche had no means of crossing water. They could not reach the mainland. They could not travel between islands in the chain. They had been on those islands in isolation for at least 2,500 years, possibly far longer, and they could not explain how they had arrived. Their own oral tradition said they were survivors of a great land that had sunk into the sea. 62 miles of open Atlantic between them and Africa. No boat, no documented origin, and over 1,000 mummies waiting in the caves. The Guanche practiced deliberate mummification. More than 1,000 mummified individuals have been recovered from cave burial sites across the islands, concentrated on Tenerife and Gran Canaria. The mummification process was sophisticated evisceration of internal organs, packing of the body cavity with specific plant materials including juniper bark, pine needles, and helichrysum flowers, followed by desiccation in sun and wind. The procedure matches ancient Egyptian mummification in its essential steps: evisceration, botanical packing, controlled drying. Egypt and the Canary Islands have no documented contact. The Guanche had no writing system. There is no known mechanism by which Egyptian mortuary technology could have reached an isolated Atlantic island population with no boats. The genetics produced their most striking finding in 2017, when Rosa Fregel and colleagues published ancient DNA analysis of 48 Guanche individuals in scientific reports. The dominant mitochondrial haplogroups recovered U, 6, H, 1, J, and T were consistent with North African Berber ancestry, which aligns with geographic proximity. But the analysis also identified a haplogroup designated U6B1. This haplogroup exists in the Canary Islands. It does not exist anywhere else on Earth. Not in North Africa, not in Europe, not in any population that could plausibly have interacted with the islands in any direction. It is a genetic line that began and ended on those 7 islands, a branch of the human family tree that evolved in total isolation long enough to become unique and was then effectively erased within a century of Spanish contact through conquest, disease, and forced absorption into the colonial population. Physical analysis of skeletal remains in the Museum of the Canaries in Las Palmas has produced consistent measurements of unusual stature. Multiple individuals show reconstructed heights between 6 feet 2 inches and 6 feet 8 inches. The contemporaneous Berber populations of North Africa averaged approximately 5 feet 5 inches. Colonial Spanish sources consistently describe the Guanche as tall and physically powerful, and the skeletal record agrees. On the island of Tenerife, a series of stone pyramid structures, the pyramids of Guimar, align precisely with the summer solstice, sunrise, and sunset. Norwegian explorer Thor Heyerdahl investigated the structures in the 1990s and concluded their astronomical alignment was deliberate, consistent with construction principles found in both Mesoamerican and Egyptian step pyramids. No mainstream theory adequately explains how an isolated Atlantic island people, with no boats and no documented external contact, produced astronomically aligned step pyramids, consistent with traditions on 2 other continents. The U6B1 haplogroup exists nowhere else. A people appeared on islands they couldn't have reached, built structures aligned to the same astronomical events as civilizations they never contacted, preserved their dead using techniques matching a culture 1,500 miles away, and carried a genetic signature that has never been found anywhere else in the world. Then the Spanish arrived, and within 100 years, the Guanche were gone. From the Starchild skull in a Chihuahua storage room to mummified giants in a Nevada cave, from Sumerian tablets describing genetic creation to a ghost population mummified in a Chinese desert, from a 6-inch skeleton with 64 unknown mutations to an island people, no boat could have delivered 10 separate remains from 10 separate civilizations across 6 continents and 7,000 years of history. Each one breaks a different rule. Each one contradicts a different piece of what we think we know about where we came from. The official explanation for each is technically possible and deeply inadequate. And in the cases where the physical remains could settle the question, they are restricted, repatriated, looted, or classified. The pattern of the anomalies and the pattern of the missing evidence point in the same direction. If this raised more questions than it answered, that means we did our job. Subscribe for more. Drop a like if any of these cases hit differently. And tell us in the comments, where in the world are you watching from?